histology of retina. Histology of retina is important because recently, if you are preparing for your NEET PG entrance exams, they give you a slide and they ask you about not to identify this, rather they will ask you like what is this layer. So, and let me tell you that this everything will be not there. So this is the way they will ask you that identify the layer of the slide, right? So first of all, they will not even mention if it's a retina, just identify the layer. So one thing is you should know how to identify first of all that it is a retina, and then how to identify the layers. So we will do this now. As I told you before, to identify a slide of retina, you should be very clear about two things, that on the inside it will be a clear cut. It means clear cut means like simple. This is the inner limiting membrane. You just have to look about these few structures to identify retina, that on the inner limiting, on the inner side it will be clean because it's filled with vitreous humor. So inner limiting membrane will have no connective tissues, no fibers, nothing attached to this. While on the outer layer, it will be lot of connective tissue, blood vessels, capillaries attached, and because it is choroid, this is choroid. So on outside this will be so that means you have this idea that it is retina. And another thing is there will be 3 layers of nuclei, 1, 2 and 3. And then I told you that retina is made up of 3 set of neurons arranged in a linear arrangement. So you have 3 neurons, so that means 3 layers of nuclei will be there. Okay, so that was about the identification of retina. 3 dense packed up layers of nuclei, one thing. Then on outer and inner side, on the inner side it will be the smooth margin in limiting membrane, on outside it will be connective tissue attached. This I hope will help you identify the retina. Now to for the counting of these layers and to identify the layers, as I already told you that the choroid side is the outer side, and from where you start numbering them. So there you find that this layer of epithelium, this is the pigment epithelial layer, the pigment cell layer, and the red color line is the Bruch's membrane. So this is pigment layer. Then this is the red layer of the photoreceptors, rods and cones. This is the layer of this is called an external limiting membrane. Then down below, and then you have this outer nuclear layer, which is formed by the cell bodies of rods and cones. This is the outer plexiform layer, which is the axons of rods and cones. Then you have inner nuclear layer, as well as you have horizontal neurons in this layer, and you have amacrine cells also, Muller cell bodies also here. This is the inner nuclear layer. Then you have inner plexiform layer. Then you have the Golgi cell layer, the nerve fiber layer, and this is the way. So one thing, identify retina because of the 3 bands of nuclei, and to start counting you should know that the connective tissues attached side is number 1 and the smooth clean margin is the number 10th layer. Okay. So that was about the histology of retina. Now this is about the difference in the histological sections of central human retina. This is a section from the central human retina, and this is from the peripheral human retina. Okay. So now let's find the differences in the central and peripheral human retina. Just keep thinking what I am telling. Like as you move from the periphery to the center of the retina, the thickness of the retina keeps increasing up till the margins of macula, because macula itself a shallowness or a sunkenness or a depression. So, from the peripheral margins of the retina, as you converge towards the macula, the thickness of the retina keeps increasing. That's number 1 point. Then as you move from the periphery of the retina towards the center of the retina, the cones keep increasing, the rods keep decreasing. Okay. Now to identify the and compare the two, right? Peripheral and central retina. Keep on looking here. On this side, you see is the layer of, although they have written rods and cones, but this might be few rods in macula, periphery of the macula, but in the fovea centralis you'll have only cones. Okay. Then there you find is the outer limiting membrane, and this is the outer nuclear layer, and this is the outer plexiform layer. So can you see in a broad way, the major difference, as I told in the very beginning? Can you appreciate this obliquity of the cell bodies of the cones, right? And these are the cones, the photoreceptors, they are also obliquely placed. Got it? And can you see the axons? The axons of these will also be obliquely placed. I hope you are getting my point, what I was trying to say when I was teaching you about the fovea centralis. Like removing the veils of the bright from outside to inside, and that's how you create a shallowness in the macula. So because of those macular slope, these axons are obliquely placed. So this is one feature. Can you appreciate that there are obliquely placed neurons in the outer nuclear layer? This is one feature which will help you identify the central and peripheral, because here you find is the vertically aligned rods and the vertically aligned nucleus of these rods and cones. Okay, so that was one point. Then important point is here now. Look upon this point is, let's use some different color. Yes. So these axons of the rods and cones, when they reach here, right? First of all, look here, there is some line here. There's a faint, pale band here, pale line here between the outer plexiform layer and inner nuclear layer. So these axons of the rods and cones, right, with the help of these cells. Remember what was these cells? These are the Muller cells. So with the help of the processes of the Muller cells and the axons of the rods and cones, they form a layer here, a band here. It's a faint fibrous band in between the outer plexiform layer and the inner nuclear layer. This faint layer, you can see this faint layer, you just remember it as this is called very important, Henley fibrous layer. This is Henley fibrous layer, which is a hallmark feature for identifying for which actually is not present. This is Henley fibrous layer is not present in the retina, in the peripheral retina, while it is present here in the central retina. Got it? And Henley fibrous layer is formed by the axons of the rods and cones with the processes of the Muller cells. This and it doesn't take good stain, so it's a pale fibrous layer called Henley's fibrous layer. Got it? So this is number 2 point of identification. The third point of identification is also very easy. Third point of identification, look here, just you have to look on these 3 layers. Can you see the thickness of these 3 layers? And can you see the thickness of these layers? Got it? So the thickness of inner nuclear layer, inner plexiform layer, and the Golgi cell layer, right, and the nerve fiber layer, all the layers will be much more thicker in central human retina. Remember, we are talking is about the from periphery to center, but at the margins of macula. Okay, then these piling of these neurons here, that is making it so thick here at these 3 layers. And the reason is of the thickening here is that apart from obviously in the peripheral portion you have more of rods, either you have more of cones is one factor. Then the these cells I'm talking is the bipolar cells and the Golgi cells. The bipolar cells and Golgi cells, both these cells are more in number, more in packing density in the central retina. One thing. And if cells are more in number, obviously their plexiform layer will also be more, because more of cells, more of axons, right? So that's why these plexiform layers are also, this is the layer of the bipolar cells, inner plexiform, and Golgi cell has this inner, they have not written it actually here, that is the inner nerve fiber layer. This they haven't written is nerve fiber layer. Okay. Here also it's the nerve fiber layer. All these layer will be thick in here. The important thing is about the impulse convergence. Let's know about this impulse convergence, because it's talking is about here. Yes, the bipolar neurons are more. All bipolar and Golgi cells, both of them are more in the central retina. So if bipolar cells are more here, right, and there is only or less of rods and more of cones here, right, and here it will be very much populated, this layer, the outer nuclear will be densely packed with the rods. And in the very beginning I told you that, remember, there were 125 million rods in a single eye and only 7 million cones. So that means in the peripheral retina, if you're taking a section, the population of rods, it's very much high, right? And the thing the other ways, now these bipolar cells and Golgi cells, they are less in number compared to the central retina. So what is happening, you can remember is that the axons of the cones, let's say you can remember this way, that convergence means like 1 or 2 or 3 cones will anastomose. Actually there is less of space, instead of drawing you just keep listening, that will be better. So stimulus convergence के बारे में we are talking about that let's say 10 to 20 cones will synapse with a single bipolar neuron. Here 10 to 50 will synapse with a single bipolar neuron here. This is a different cell, right? These are the cones which anastomose here. And think here in the opposite side on the peripheral retina, what is happening on the peripheral retina, there are rods, and rods are very much high in number, right? So there will be like hundreds of rods converging over a single bipolar neuron, right? So there is more convergence of stimulus, and this will be less convergence of stimulus in the central retina. 10 to 20 cones will synapse with single bipolar. Reason is there is so much of bipolar density is much more here, and the cones density is number of cones compared to the number of rods here is very less. So that is how the convergence in this peripheral retina will be very much more. And the rest of them you have already told you just simply have to see that even if this ganglion cells, if will be more in number, there will be more of nerve fibers, because the nerve fibers are actually the continuation of the axons of the ganglion cell layer. So these are the few points I believe you must have understood, that how to identify the central human retina. The point number 1 will be obliquity. The point number 1 is obliquity, obliquely placed the cones or their nerve fibers, they will be obliquely placed, right. And point number 2 will be the Henley fibrous layer, number 2. Number 3 will be the thickening, of these layers, and fourth will be the convergence. You can just remember like this, that the visual impulse convergence is less here, it is less here, and this fourth point is more here. These are the points you can remember that will help you differentiate the central and peripheral human retina. Okay.